Good evening, everyone. Welcome back to NPTEL live session for transition metal organometallic course in chemistry and biology. So today we will be taking up the topics that have been covered in week 4 lecture series, and at the end we will be solving the assignment questions. But if you guys have any doubt regarding what we have learned in the previous weeks, please feel free to hello. So if anybody is having any doubt, they can unmute yourself, and or you can put a question in the chat box. So we will wait for 2 to 3 more minutes for others to join, and then we can start with the lecture.
वही जून के 9।
Okay, so we can start the today's session. So these are the topics that I've written onto the first slide that we are going to cover today. Firstly, we will be talking about different types of Gibbs catalysts, like different generations. So we will try to understand the mechanism, how they are actively participating in olefin metathesis. Then regarding that, we will get to know about the influence of the steric hindrance and electronic environment around the metal center. Then the next would be the hormones comparison study regarding the Gibbs catalyst 1 and 2. After that, we will be having alkene metathesis catalyst. We will take it into more detail. Further, we will go for cross metathesis and then SHOP, which is shell higher olefin polymerization. And lastly, we will be taking up into consideration the limitations of cross metathesis. So let's start with the first topic. So in the last live session, we were talking about olefin metathesis catalyst. So here, what was the metathesis? Say if I'm taking these two are different catalysts, olefins in presence of the catalyst, they would be forming this product and ethylene. So R1 and R2 they are not same. That's why we compare that this one olefin, it is reacting with other olefin, giving us a new and third olefin. This is new olefin, which is our product. And here when we are saying catalyst, with the catalyst we mean, and we saw in the last sessions that we were talking about the metal carbene catalyst. And in that, we have seen we have two types of metal carbenes. One is Fischer carbene, second is shock carbene. If I will say this is your singlet carbene and this is your triplet carbene, we have already seen here. We have, if we'll go back and see the live session of the previous week, we will get to know about Fischer and shock carbene in full details. So here the point is about the catalyst that we are talking about, the metal carbene. And in that one, what we said is this metal carbene. Here the metal could be your tungsten, rhenium, molybdenum, and ruthenium. All are noble metals. But among those, the most used and most active catalyst that has been seen, that is your ruthenium based catalyst. Why? The advantages that we have if we have ruthenium based catalyst is that they are easy to handle. Handling is easy. These complexes are easy to be prepared. That means synthesis is easy. And most importantly, they have functional group tolerance. That means even if ruthenium is having a carbene like this one with other substrates, this complex is not going to be affected by in presence of any other functional group. Say for example carbonyl or NH2, etc. Any functional group, if you are putting into the reaction scheme, they are not going to react because they are that much tolerant. So this is a highlighting feature of the ruthenium-based metal carbons, and this is what our main topic is, that if you are considering the ruthenium as our metal center, ruthenium as our metal center, so we have in general Krebs catalyst, where if your ruthenium is bound with phosphine groups and chlorine groups with pH carbon. So this is your first generation Krebs catalyst. Then if I am replacing this 1PCY3, that means tricyclohexyl phosphine group, by an imidazole kind of system, we're having these groups attached. Methyl groups attached, and this is further connected to PCY3 double bonded. So this is your second generation catalyst. Here we have another version of it. Indium, which will itself acts as carbon, and the same group which we are having earlier. So this is the unsaturated version, where we are using the N-heterocyclic carbon, and so on and so forth. So we have these kind of systems which come under the Krebs catalyst, and they participate in orbital metathesis. So then, how these things work, the mechanism of a Krebs catalyst in a metathesis reaction. So this involves PCY3 dissociation and olefin binding, the two major steps. So, for example, we are taking ruthenium, chlorine, PCY3, PCY3, double bond H, CH2H. Now here, this first group will leave. This is called PCY3 dissociation step, which is kind of very fast step. And what it will do, it will create a vacancy. This is your vacancy. Now, if I will introduce an alkene over here, at this frequency it will start going to have an interaction. So this is now we call as Cl renal, this is by P, this is on pH, binding with your, so this is called a olefin binding step. Hereafter, we will be having an intermediate, which we have already discussed, metallocyclobutane kind of intermediate, given by Chauvet and iron chloride and other groups are as such. And then it will give this kind of aniline and this kind of aniline, this kind of structure, and ended up having a, it will give you new metal catalyst as CH2, PC3, Cl, NCl plus pH and 1. This is your product olefin. And this is your newly generated metal carbene, which can again be reacted with another molecule of olefin to give you the corresponding products. What happens here? The PCY3 step is faster. If it is faster, then only we can have an olefin binding very easily. So how and what are the ways to make these steps a bit more faster to have our product? So we are talking about different ways of increase the activity of catalyst. Specifically, if I would say Grubbs catalyst in olefin metathesis. So the first one is to increase the phosphine dissociation rate, which can be done by replacing the PPH3 group by PCY3 group. That means with more bulky ligands. Also, by replacing with phosphine ligands having more bone angle. Also, we can replace the other substituents for other groups, say chloride by iodide. So more bulkiness. And the second point is to have more electron donating phosphines, because more the electron density going to metal to sigma bond, it will donate the electrons to olefin by metal living pi donation. This is a very important step over here. So the two major features that we are going to look into that we talked about today is how we can increase these two steps which we were showing in the mechanism. That first step to take out this PCY3 group to make a vacancy so that this vacancy can only make your olefin to come nearby to your refinery. So the first step is has to be quickly should dissociate. That can only be possible if we have a bulky group, they are good leaving group, so they will go away. Also, the iodides would make it sterically hindered to go easily. And the second phosphine group should be giving electron density to the metal center so that this would become metal electronically rich and that could tie back do the back donation to the olefin and therefore can interact with the olefin and make the binding more stronger. So these are the two further steps which are required to fissile the olefin metathesis catalysis. Now to understand it more, understand it more, if we are already so now taking an example, if PCY3 group in Krebs catalyst, which is first generation, is replaced by an N-heterocyclic group, which is your Krebs second generation catalyst. Then what will happen? So if we have this particular thing, so what are we trying to do? We have the ruthenium. Here it's PCY3, it's PCY3 chloride, second chloride, and your carbon. And if we are replacing this group with your N-heterocyclic group, here, save, and PCY3, double bonded pH. So when we are doing this kind of thing where this has been replaced by this whole group, so we get to see more activity with this. How? Now when we have, I'm just writing pH dash here equals to this group 135 trimethyl benzene group. So I'm just writing here. Like if I am having this ruthenium, the same heterocyclic group, which is having pH dash, here Cl, PCY3, double bonded pH. Now, first step would be very fissile to give out your PCY3 dissociation. Why? Because this is bulky group also, as well as electron donating. So when it goes away, it will form a vacancy over here. And then this becomes coordinatively unsaturated. Now it has space to coordinate another thing. Then what it will do, when olefin will come into picture, and here is the ruthenium, it will start having an interaction with olefin, the second carbon over here, and here. groups are attached. H dash, H dash, C. Now here this NHC group is a very good sigma donor. So it will make a very strong bond. Here the sigma, if I'll just mention over here, this donation would be very strong and more electron density on ruthenium. Therefore, ruthenium to olefin by back donation would make olefin binding more strong. So this then becomes very strong, and we ultimately will be having Cl, ruthenium Cl, and R, pH, and N, and then this can break, and you will get your product pH and R. This is your product. So this way we can, so here by replacing the, so by replacing PCY3 with N heterocyclic carbene, which is Grubb's first catalyst. This is Grubb's second generation. Here, this is more sigma donor, more bulky also. So first step of PCY3 dissociation becomes faster, and more sigma donor makes olefin binding faster. So overall, catalyst activity increases with this one. I hope this is clear to everybody. Again, to compare these things, to compare the activity of Grubbs 1 and 2, Osman named scientist did a demonstration. So he actually compared the activity of both G1 and G2. G stands for Grubbs catalyst in ROMP, which is ring opening metathesis polymerization. So this is your ring opening metathesis polymerization of cyclohexene. Please remember this name, it's very important. So he actually compared these two catalysts for the cyclohexene ring opening metathesis polymerization. So we will see here. So here we are taking cyclohexene And in presence of our catalyst, we could open up this chain. -25°C. So here cyclooctane is 2. The catalyst amount that has been taken is 250:1. So what kind of graph that has been seen, that if we are talking about the percentage yield of our product, so when it was the Grubb's first catalyst, first generation catalyst, where both the substrates are PC/3, so the graph was going like this. So the graph was going like this manner, very steadily increasing. But when it was about the Grubb's 2, when it was being replaced and did the reaction with Grubb's 2 catalyst, the graph reaches to here. So this is for the second generation. So you can see the high percentage yield for Grubb's 2. So this was being demonstrated in Hammond's experiment of cyclooctane in ring opening metathesis polymerization. I hope this is clear. Now we will move on to our next thing, which is alkene metathesis catalyst. We will take up this into more consideration. The topic is alkene metathesis catalyst. We have already talked about that in the metathesis catalyst, we are talking about this carbene, where M stands for molybdenum, tungsten, rhenium, ruthenium, and ruthenium is found to be the best among them. So, say for example, we are taking for how it is being made. So we have taken tungsten chloride plus dimethyl aluminum chloride. This reaction has been asked in your last assignment. So under this picture, 2Cl will be replaced by your methyl groups, and here we will get chloride, CH3, CH3, and then we are treating it with ethanol with the loss of water. So this will become WOCl with oxidized, and the CH3 that we are having will start breaking up the bond with tungsten, start making up the bond with hydrogen, and here there would be two bonds with the CH2, that CH3, which is having already the bond with your hydrogen. So it will start breaking and it will start making. So here, this will leave as your CH4, and here it will form C double bonded at CH2 and Cl and Cl. So this is your catalyst for metathesis reaction. Similarly, we have a lot of Grubb's catalyst, and so we will just see what are the advantages and disadvantages with different types of metal carbon complexes. So if we are talking about the shocks complexes, which have been given in 1990, what kind of complex we have? We have the preventive molybdenum. This is an amide kind of system, which is having diversified group. And here we have the preventive page. This is H. And there are two similar groups. We're having CF3, CF3, and methyl. Similarly, here oxygen, CF3, CF3, and methyl. So these groups, which are the CF3, CF3, the electron withdrawing groups, so the presence of the electron withdrawing groups onto the metal itself, it increase the catalytic activity and also it even let the sterically demanding olefins, say tri and tetra substituted, that also can undergo metabases. So these are the advantages of it, but the disadvantage that I'm highlighting there. So the disadvantage is that poor functional group tolerance. Whatever we wrote over here, these were the advantages with Short's catalyst. First is this one, second is this one, and this is the disadvantage with the Short complexes. So when we are talking about another group, Grubbs catalyst, so another metathesis catalyst, Grubb complexes, which came after 5 years, 1925. So for example, ruthenium, chlorine, PC2, which is the first generation, double bonded pH. Where two fluorines are having 165, 67 degrees. Since this meeting is going to end in another 1 minute, I request you all to please join the symbol. Thank you. So firstly we'll write the advantages. So the first advantage is, as we said, that this is functional group tolerant, which was absent in case of shock carbons. Secondly, they have selectivity. So the disadvantage